Hi, everybody. Go ahead and check your audio settings, and we'll be getting started here shortly at 3 o'clock with our next LionSide Chat. In about 1 minute, we're going to get started with our LionSide Chat today. So go ahead and just make sure your audio settings are all ready to go. Okay, everybody, we'd like to welcome you to today's LionSide Chat. My name is Sonia Delacueto, and I am one of the 3 LionSide Chat moderators along Dr. Ryan Hassler and Professor Dawn Wrights. We would like to invite you to come on in and welcome to our campus. We're so glad that you're able to join us today. But before we get started, we wanted to share that you should please feel free to submit questions via the Q&A feature as we're going through our program. Once the presentation concludes, we're going to facilitate a discussion with our presenter, and we'll also be recording today's session so that you can revisit the topic or share your experiences with friends and family. So now what we'd like to do is welcome Dr. Ryan Hassler, who is an associate teaching professor here at Penn State Berks and one of our own LionSide Chat moderators. He's been teaching statistics here at Penn State Berks for 9 years, and those 4 lovely beasts on the screen are his favorites. So now what I would like to do is introduce Dr. Ryan Hassler, who is going to be speaking about the vaccine. The lovely vaccine. Where is the vaccine, Dr. Hassler? Can you help analyze some statistical data related to the COVID-19 treatment for us today?
That is the goal, so we will see. All right, thank you. Get this set up here. All right, so thank you, Sonia, for the introduction. The topic of my conversation is where's the vaccine? And although I'm discussing here statistical data, I want to rest assured that this is a very friendly talk, so we won't be diving too much into mathematics. And I also wanted to just, mention up front, I'm not a medical doctor, so what I'm sharing with you here are just the results of research that has been ongoing and what I have found in terms of the process of where we are currently in this search for a vaccine. So the agenda for my talk today, we're going to just very briefly touch upon what good experimental design is. We'll talk about the phases of clinical trials. We're going to talk about what does it mean to accelerate a clinical trial and the timeline. We're going to talk about the current status of vaccine candidates, lessons maybe that we have learned or could learn from prior searches for vaccines. And then also we'll talk about therapeutic treatments to round out the talk. So just to make sure everybody's on the same page, in terms of what a clinical trial is, let's first define an experiment as a series of observations that are made under conditions that are controlled by a scientist. Now, when we apply medical treatments to the human body in this experimental process, that is what we are calling or defining as a clinical trial. So an experiment that is looking at medical treatments on the human body or on subjects. And before we go too much into detail here about where we are, I want to just put up a couple quotes here to remind you that the best time to contemplate the quality of evidence from a clinical trial is before it begins. So if we want to make sure that our results are both effective, accurate, precise, great care typically needs to go into the setup of a clinical trial. And that I think is captured very well with this next quote, but keep these in mind as we talk about, what is happening in terms of the acceleration of the vaccine search. So in general, high quality evidence about the effects of new treatments is a consequence of good study design and execution. The good study design, of course, is before the study happens. The execution is what happens as we are, actually going through the clinical trial. And all of that is a result of careful planning. So I just wanted to start the talk off with those, with that sort of preface in terms of what is needed for good results or for effective results at the end of a clinical trial. And then most of our talk is going to talk today about vaccines. We will mention about therapeutic drugs at the end. Just want to make sure everybody's on the same page with this as well before we begin our conversations. A vaccine is more of an imitation of an infection. Of course, I think most people can understand what it means in terms of vaccines that are applied to their body. But the idea of a vaccine is that we vaccinate so that the body has a memory about what that disease or the virus looks like, thereby when the body would get exposed to it, it would be reducing the rate of infection. So vaccines, of course, are with healthy people, versus a therapeutic drug is really a drug that's used to treat when somebody actually has the virus. And then, of course, we're looking to minimize the effects of the disease or the virus. So we're going to talk primarily about vaccine and the search for that, but we don't want to remiss and not talk about therapeutic drugs, which we'll mention towards the end. So to start off, let's just real briefly, and again, I could teach an entire course on experimental design, multiple courses on it actually, but really briefly to just sort of make sure we're situated with some of the verbiage that you might see. A good experimental design includes randomization. Now, what do we mean by that? Randomization uses chance to divide subjects into treatment groups. And really the purpose behind needing to do this is to remove bias for uncontrollable variation. What we really want to do is be able to spread this uncontrollable variation equally across groups. So what does that look like? So let me give you a quick example. Let's say we have 8 subjects and we want to divide those into 2 groups, the treatment group and what we're going to define in a minute as the placebo group. But let's say that 4 of those had a pre-existing condition. Let's say, for purposes of our talk, let's say 4 had high blood pressure or 4 were diabetic. Maybe a pre-existing condition in which we know that COVID-19 affects people differently and, of course, negatively with those other conditions. If we did not do randomization, those 4 of the 8 could potentially end up in one group. And if they ended up in one group, such as I showed here, then we don't know necessarily if the drug maybe that we're giving to treat them is as a result of the pre-existing condition or as a result of the actual treatment. So with randomization, I know it's really quick here, and of course we use more than 8 people, we'll talk about that, but the goal would be to make something that looks more like this, that all of those uncontrollable variation or differences among people when we go into a clinical trial would be spread evenly across all of our treatment groups. So in this case there's 2 that would have the pre-existing condition in both cases. So randomization is important. The next thing you're going to hear is important is the idea of a control or a control group. And a control group in the medical field typically is the idea that one group is not receiving anything, and we typically call that a placebo when we're dealing with clinical trials. And the purpose of this is to compare the active treatment to the status quo or to a group that would get nothing. And that is really for the purposes of seeing how effective the drug is. And I just want to remind you, we'll talk a little bit more about this later as well, but uncontrolled studies that don't include placebos in medicine often give new medicine much higher success rates than those that actually are comparative experiments. And just wanted to mention real briefly, because you maybe have heard of these 2 things. First, the idea of the placebo effect. Use of a placebo or a fake treatment, somebody that gets nothing, allows researchers to isolate the effect of the study treatment from the placebo effect. And the placebo effect, what's shown there on the picture, if you believe something will affect you, it probably will. And if you don't believe it, it won't. So what we're trying to do is combat this idea that, people psychologically are reporting positive or favorable results, that it might not be due to the treatment, but might be due to their psychological, beliefs about them being treated. Another thing that's important is we typically want to blind our subjects, and oftentimes you might hear double blinding. That's where the person who's administering their drug doesn't know which patient is getting which drug, and the patients also don't know. And again, all of that is to make sure that we're preventing the researcher from treating the two different groups differently, and/or having the patients know which group they're in. So you'll hear about randomization, you'll hear about control, and then the third principle we typically talk about is replication. And so with replication, subjects can't be 100% identical. Even though we might try to make our groups as similar as possible, the end result is that you and I are different. Are those differences that we might see in our results at the end of our clinical trials, are they a response due to the treatments that we're actually applying, or could that just be due to random chance? And of course, we're not going to dive too much into the mathematics, but that's really where the mathematics comes in, is due to treatment or due to chance. And therefore, large numbers of subjects, right? We can't just do clinical trials on 2 or 3 people, but large number of subjects are going to be needed to decrease what we call experimental error, and ultimately gives us precise estimates of the treatment effect. And so let me just, again, frame this as an example. So if I ask you the following, let's say I'm going to flip a coin, and I'm going to ask you, is my coin fair? And my first example for you is I'm going to flip that coin 4 times. And you can see the results there on my screen of the 4 flips that I actually have. I have 3 of the 4 turned out to be heads. And I know we're not too interactive here in this setting of a webinar, but if I asked you during my class, is my coin fair? Right now, you should say yes. Now, you might say, well, it's not 50-50, but you don't have a whole lot of evidence to suggest that my coin is unfair. Even though 75% were heads, it's only 3 out of 4. So I would want you to respond that, yeah, your coin is most likely fair. But if I flip that coin 100 times, and now I get 75 heads, versus 25 tails, I still have 75% of my data being heads. But at this point, you should tell me that my coin is not fair, because getting 75 out of 100 versus 3 out of 4, even though the percentages are the same, really means a whole lot different, really means a different outcome. And really that's because we have more information. And in statistics, that's known typically as the law of large numbers. So with randomization, with control, with replication, with proper study design, what we can see when we are going through clinical trials is where, my discipline of statistics comes in, statistical evidence. And again, without going into the math, statistical evidence is when an observation that you see in as your results, when you're analyzing your data, is so different than what you expected that it rarely could occur by chance. There was no way it actually happened by chance. And if it didn't happen by chance, it then must be due to the treatment that you're looking at. And that's where we are implying our efficacy. So let's move here now into what's a clinical trial. What are the stages of a clinical trial? And as I go through some of this, I might not, read out loud everything that's on the slide, and you can, kind of scan it as we go. But the first stage or the first phase would be a preclinical. This is actually usually done in laboratory settings and with cells or animals. Typically, or actually, this does not include humans. The goal of this, prior or pre-stage is really to look at research in an exploratory setting. Here are some questions that we might ask or investigate. Is the drug that we're looking at harmful to living tissue? Not necessarily harmful to a human being because they're not in a human being yet, but to living tissue. What more needs to be known about the chemical makeup, perhaps, of a drug? Or what substance of the virus might produce an immune response? So this is the preclinical trial. Once we have a fairly good understanding of the exploratory analysis of what we're dealing with, the very first stage of a clinical trial would be called phase 1, typically up to 20, 30 subjects or so. And as we go through here in a therapeutic drug, when we're not talking about vaccine, we typically are going to apply this to somebody that's sick and maybe has very limited options for how to get treated. Versus a vaccine, of course, we're using this in healthy subjects. The goal of a phase 1 is safety. And so questions we ask here: what is the best dose? What quantity, what frequency should we apply this drug to? Is it safe? What would we start to look at? What are some of the very beginning side effects that we might see? If it's a vaccine and we're, putting it into as an injection, well, that's my next question here. Do we do it as a pill or an injection? And if it's an injection, maybe do we have adverse, events? Is there a localized, pain where the injection was at? Phase 1 is a safety phase. Once we can show that there's really not a whole lot of harm, we move to phase 2. This is just an expansion of phase 1. And the idea here, especially, is to kind of go a little bit further and look at the efficacy. How good is the drug or the vaccine working? Of course, in the vaccine, we really want to start to move towards applying this to a demographic of people that we might eventually be putting this, worldwide or countrywide spreading distribution of the vaccine. So we might try to, match the population. Questions here for efficacy: what are the most common short-term side effects? Now that's sort of the safety as well, but we continue to monitor side effects as we go through all phases. Really, how are the volunteers' immune systems responding to the vaccine? So really, what is actually happening in terms of, the drug or the vaccine process? And then once we get through those 2 phases, we move to phase 3, which this is really the biggest phase. This is the largest phase and the last phase before, you would get to approval through the FDA. And this is often tens or hundreds of thousands, or even more subjects. So this is a widespread, large distribution of the drug or the vaccine. And the goal here, yes, safety, yes, efficacy, we're still monitoring, but the goal is to put into a comparative statistical evidence. Let's compare what we're giving here to either the placebo and the treatment group or the status quo drug versus what our new drug is. So we're looking for how do people get the treatment vaccine compared to how do people do without it. We're looking at, do the previous safety results still stay true, or do we have, more information here that suggests maybe it's not as safe? And then the last one would be phase 4, and this is after approval, and this is, the ongoing research in terms of looking at long-term side effects or long-term safety. So really, approval comes between phase 3 and phase 4. All right, here's a typical timeline. So now we're going to talk about the timeline of a vaccine process for successful clinical trials. Couple things I want to point out here is these are just, I pulled these are typical timelines. This was from the FDA website. Typical timelines of how long it takes to move a treatment or a vaccine through the phases of the clinical trial. And then over here on the right side are the probability of success. So a lot of drugs don't even make it out of preclinical trial, but then if they do, only about 65% make it through phase 1, 50% of those 65 make it through phase 2, 60% of those 50% make it phase 3. So again, I'm not going to do a whole lot of math here. You start to compound and multiply those numbers together. This is taking many years typically, and only about 10 to maybe 15% of drugs that start out in the preclinical phase will ever make it to market. And so here's what it looks like, just broken down a little bit further for different, different types of diseases. So we got oncology there, cardiovascular, infectious disease. I'll highlight just this one here, the overall. And so you can see overall, about 13.8% of trials that actually start in the preclinical phase actually will get to marketplace. And so what does that mean? Here are some other examples of historical vaccine timelines. Again, we're talking about the timeline. What does it mean? What's traditionally happens? And so there's just a couple examples. The first one there is the chicken pox, 28 years it took to develop the vaccine. The next one is your typical influenza, the 28 years as well. And then here we are down here, our goal as a world, as a country, whatever, whoever you're listening to, we're looking at maybe 18 months. And so what are the implications of trying to get something in such a short amount of Let me talk about now how clinical trials currently are being accelerated. And sort of, Dr. Peter Hotez, which is from, the dean down in Texas, the dean of the Baylor College of Medicine, probably, puts it the best. If you want to make this 18-month time frame, put all your horses you can into the race. So, that means money, that means, investments, that means worldwide research. And so, that's sort of the forefront here, is that we have the world working on this. But here are some other ways that we are accelerating this process. First off, COVID-19 is about 80% similar to SARS and MERS in terms of a coronavirus. And so we have a ton of prior research, although it's not on this novel COVID-19, we have a ton of research on SARS and MERS. And so we have been able to accelerate that preclinical trials. And this is actually what has led us to a rather quick test. A test to determine if somebody is positive for COVID-19, because we had a lot of research behind coronaviruses. And I know, one of my colleagues has talked about that on the LionSide Chat already up to this point. Also technology, 50 years ago, we were recording everything paper and pencil in clinical trials. Now we have almost instantaneous access. We're recording information on tablets that get almost instantaneously uploaded. And so in terms of the record keeping, technology has really helped and will be able to help us accelerate. We also have what's called emergency use authorizations. We'll talk about that a little bit more with the FDA. And so what that is where we can maybe skip a couple steps and use an emergency authorization, and we'll mention that in a couple minutes. We also, the FDA has an immediate review of study protocol. This is a big one here. We're going to overlap phases of the clinical trial, so we're not going to start a new phase right when the last one stopped. We're going to have some overlapping here, and we are going to try to accelerate that phase 3. That's really the biggest phase, the large-scale testing, sooner. Perhaps we can build factories so that we can get ready to manufacture the vaccine even before we know the vaccine will work. And then lastly, we also are looking at, technology and experimental research in terms of RNA is a type of new vaccine that we are hopeful for. And if we can, find an RNA vaccine, it might be faster to produce and distribute. So there's, a lot of things in the game here in terms of how you might accelerate a clinical trial. But in general, the long story is it looks like this. At the top 10 to 15 years through those processes, and it's more sequential. And what I'm highlighting here in the red circle is that we're really going to try to overlap a lot. We're going to try to overlap clinical trials, and we're going to try to overlap the FDA process of approval along with that rapid manufacturing. Try to get ready to do the manufacturing before we might even have the vaccine ready. So where does the world stand? Let's now talk about where are we in the search for this vaccine. So as of July 31st, so a couple days ago, the end of July, we have 200 plus vaccine candidates. We call them candidates because, of course, they might not become the vaccine. Most of them won't, in the preclinical development stage. So, maybe not into humans yet, but we're, analyzing, in the laboratory setting. We have 24 current vaccines, and actually, I think I heard a report this morning that said 26. So as of 3 or 4 days ago, there was 24, and something I heard this morning said there were 26. So this is going to continue to increase as those preclinical trials start to show some maybe positive results in the animals we're using or the cells. We have 300 plus therapeutic treatments, so not actually vaccine, but treatment for COVID-19. And so the world is, very much all working, not necessarily collaboratively, but everybody has a stake in this game. What are the types of vaccine candidates? So let's just real briefly, there's a lot on this slide, but I just want to make sure I talked about this because I'm going to mention what the US is doing. And so it gives you sort of a frame of reference. So we have really 3 different vaccine types. One is a live vaccine. This is where the vaccine is using the actual virus, but a much weakened form of the virus. And the idea there is that a pro there as listed. It's a very familiar. This is what we use for measles, mumps, smallpox, and chickenpox. Versus an inactivated vaccine, which is where we kill the virus, and then we use the killed virus to create the vaccine. And examples of that are flu, hepatitis, and rabies. So these are our two, types of vaccines that up to this point in our world that we typically have approved for different diseases. And then the third one, which I alluded at earlier, is this idea of a genetically engineered vaccine. The idea that maybe we can use RNA and DNA to provide our bodies with instructions to prompt the immune system for antibodies. So the messenger RNA, as opposed to actually putting an actual vaccine, whether it's or an actual virus, whether it's live or killed into the body, maybe we can come up with an imitation using messenger RNA. And so that's very much exploratory, but we'll talk a little bit about that here. So we talked about where the world is. Where does the US stand? And so the current Trump administration has deemed this the Operation Warp Speed. So if you hear that, Operation Warp Speed, that's this idea of let's try to accelerate the US development of the drugs. So accelerate development, accelerate manufacturing, and then of course distribution of COVID-19 in terms of both vaccines and therapeutics is the goal of Operation Warp Speed. One of the goals is we were trying to aim for 300 million vaccines by January of 2021. And so that's, 6 months from now is the goal of Operation Warp Speed. The US Congress has invested $10 million or even more into this operation. And the US government specifically is really looking at it has funded and really is focusing on 3 different candidates. So we said there's 24 virus candidates worldwide, or 26 that I had heard this morning, but the US government is really looking at 3 of those. So here are the 3. I'm not going to read all of this here. I will provide these slides on our website for you to go back and look at, but I just wanted to touch upon the 3 because maybe you've heard of these companies in the media. So the first one is Moderna. This is an RNA. So remember, there is no RNA current vaccines out there, but this is an RNA. It's really based on the SARS and the MERS studies that are similar to the coronavirus. You can just see a couple things I highlighted in yellow there. The phase 1 started late February, showed positive results for having more antibodies than recovered patients. There were, mild, no serious, but mild side effects in 50%. When we went to phase 2, 600 US subjects were looked at, and the FDA did review this vaccine and suggested to move it to phase 3. So late July, so we're talking a week or 2 ago, actually maybe it was last week if you were following the media, you probably heard a lot about Moderna, the very first stage 3 clinical trial in the US. It's currently recruiting 30,000 subjects at high risk places and 90 locations across the US. And the goal, according to, I'm just pulling this from the FDA website, is to follow those people in this phase 3 for up to 2 years. So 6 months from now, the goal is to have a vaccine, but according to the FDA, we're going to follow stage 3 here for up to 2 years. So that's the first one that the US government has invested in. The second one is AstraZeneca. This is a live vaccine, so different than the messenger RNA. It's actually a weakened cold virus from chimpanzees. Stage 1 and 2 trials were kind of put together. This was done in the UK. Single doses were 4-fold increases in antibodies. So really it showed, much, more antibodies than, subjects that didn't have it. And you can see the stage 1 and 2 were single blinded. There were 60% mild, side effects. And those, we're talking about maybe sore throats and tiredness and nausea, nothing that seemed too serious in terms of the adverse effects. This was peer reviewed, of course, not FDA because this was done in the UK. And currently phase 3 trials have started to recruit, again, 30,000 subjects worldwide. So we're currently in the beginning stages of that. And then the last one is the Pfizer, who's partnered with biotech. This is also a messenger RNA vaccine that the government is funding. So 2 of the 3 are messenger RNAs. Stage 1 and stage 2 trial was a placebo-controlled trial, but there was actually not significant severe side effects, but the side effects were elevated at the 100 dose value here, and this was not peer reviewed, most likely probably because there needs to be some ongoing discussions about those increased side effects. So it has not yet been peer reviewed, hasn't been reviewed by FDA, but they did currently start recruiting for phase 3. So there were some, negative side effects in phase 1 and 2, but it was placebo controlled. And so they're currently looking at 120 different sites globally with not the 100 dose there, the 30 dose, and doing it twice. So those are sort of the three candidates that we're currently looking at. In terms of the regulatory approval of vaccine, when a vaccine gets approved, it doesn't necessarily mean it works well. There's benefits. The benefits really appear to be greater than the known risks. And so just to give you a reference here, when we do have an approval, the FDA in June came out and said, we need a vaccine to be at least 50% effective. And being effective means it prevents or decreases the severity of the COVID-19. The World Health Organization prefers 70%. That doesn't mean that they won't, support anything below that, but they prefer 70%. The World Health Organization does not approve vaccines. They do. They are sort of a regulatory agency where developing nations worldwide will rely on them for quality assurance. Now, besides no better alternative, why would we approve a 50% effective drug? Well, how does a virus disappear? Beyond the measures that are taken to physically isolate a virus, such as social distancing, you might have heard of this term herd immunity. So herd immunity is this idea that once a large portion of the population is immune, the disease is going to have nowhere to go because it's infected a large portion of people. And researchers currently believe that that's about 50 to 67% of the population would be needed to be infected. Two ways to gain herd immunity. The first one is natural resistance, the idea that the body actually gets exposed, and therefore we make antibodies, and there's no vaccine in that. And that's really how the Zika virus down in Brazil was ended in 2015, versus what we've been talking about here is vaccines, which is, we put a vaccine into the vast majority of people, and that's sort of how polio ended in the United States. And let's talk a little bit about polio. So we've been here before. Whether you're talking about infections of a lung sort of virus, or you talk about the idea of having to social distance, closing a movie theaters, whatever, we have been here before. Whether you're talking about the iron lungs that were used for the polio versus the ventilators that we have used this past year for the support of people who get sick. I'm not going to read the quote here, but real briefly, history of polio in the U.S. Harry Truman did, mention that it had to be a united front to, fight the war against polio. Polio, I'm not saying that it's identical to what we're talking about with COVID-19, but there's some lessons we can learn here. So 1894 was the first outbreak. Notice 18 were killed. 1952, the worst year, 3,000 or so died in the U.S. of polio. And by 1954, maybe you've heard of Jonas Salk. I hate to even say University of Pittsburgh because we're Penn State, but the University of Pittsburgh scientist Jonas Salk concluded that his year-long research with the inactivated vaccine for polio was successful, 80 to 90% effective. And notice we're putting 30,000 subjects into these clinical trials. Salk back in 1950s used 2 million children. Also notice that the last known case of polio in the U.S. Did not happen until 1979, almost 85 years later. So, to keep that in mind when we're talking about an 18-month, acceleration of a clinical trial. Here's just a picture of polio deaths in the United States. So in 1952, at the height of the polio pandemic, we had 57,000 cases, 3,100 people dead, with 0.03% of the U.S. infected. Compare that to right now, we have 4 million cases, over 4 million. This was a couple days ago, so we're pushing 5 million in the U.S., 154,000 deaths, and 1.4% of the population has been infected. That's about 50 times higher, and that's in 6 months. So one might extrapolate, even though it might not be a good thing to do, but one might extrapolate that in 12 months, it's going to be 100 times higher. And so we're talking about, yes, we're comparing some things to polio, but COVID-19 is not exactly like that. And one might argue it's much more infectious and much more, there's a lot of differences between it. But that just gives you a frame of reference about where we were at and the number of deaths we had at the height of the polio pandemic versus what we're faced with here in the United States today. So even though they're not identical, what can we learn from this idea of the polio vaccine? What can we take away? So first off, in the 1930s, we actually had 3 early vaccines that were proved to be ineffective. 2 were ineffective, 1 was actually deadly. And so we didn't just miraculously find a vaccine. There was a long history of exploration and failure before we got the polio vaccine. Once we did have the polio vaccine, the first month into the production of it, the Salk vaccine, there was a botched batch out in California that did not completely kill the virus and actually ended up killing people who had been infected with it through the vaccine. And so production was suspended. And safety standards had to be evaluated. And so when the production commenced, it was about 6 months later after, there was, evidence that it was now safe to continue the production and distribution of the polio vaccine. When the production commenced, there was turmoil. And what do I mean by the turmoil? So first off, there was a short supply. There were underground markets, price gouging. Pharmaceutical companies or investors were giving the vaccine to their own children first before it would be, accessible to other people. Was what some call political unrest. States wanted the federal government to take lead in providing an equitable distribution among the citizens of the country. In 1955, President Eisenhower did, put together and sign the Polio Vaccination Assistance Act. But pretty much what that did was that reversed what the states wanted. It actually let the states decide for themselves. There was not a more national distribution process. Now, why? What happened as a result of that? Well, as you saw in 1955, until 1979 was the last time that we had polio in the U.S. For many years following, the vaccine was largely inaccessible to mainly urban, poor, many African Americans who had limited health care. And so, this is one thing that we, might take as a lesson from the polio surge. How far have we come? Again, at the end of the day, as Dr. Fauci says, the ultimate game changer is going to be a vaccine. If we have a vaccine, that is the game changer in an infectious disease. Although we have been connected globally, that has increased the spread of COVID-19. That's sort of, a bad thing. It's really good that, we do have the global capacity, which is far exceeds what we had in 1950s in terms of the polio vaccine. And to that, the quantity, more vaccines currently in 6 months than ever for the polio are being investigated. Safety, we have vaccine approval protocol. We have regulatory thresholds. We have things in place to make sure that we don't have vaccines or drugs that get into our marketplace that will be harmful to our citizens. But we still face challenges. So what are some of those challenges? First off, even though we have a lot of information on coronaviruses, with SARS and MERS data, we're uncertain about the long-lasting immune response. We don't know. And the only way we're going to know about this for the antibodies is with time. And so we're trying to accelerate a clinical trial, but the only way we really will know is when we actually have long-lasting immune responses. In terms of time, we're unable to expose large phase 3 subjects to COVID-19. So, even though we're, putting vaccines out there, it's unethical to say, all right, you have the vaccine, now let's expose you to the virus. We let the people, go out and live their life and then try to track them to see, is the vaccine effective? And so, according to the FDA, the commissioner of FDA, they have repetitively said that they will not cut corners in their decisions for the approval process, the protocols that have been put in place. But yet still many have a concern that, even though the FDA is saying this and we have these protocols put in place, that there will be political pressure to accelerate vaccines before safety and efficacy are proven. And so it'll be interesting to look for. Once we do have a vaccine, when that does come, we also will face challenges of developing, marketing, distribution of that vaccine, and hopefully we learn some lessons from what happened in the polio distribution, so that we have no social disparities. And again, the marketing, in general, are we going to have a national campaign? That's left to be seen. There was a national campaign for polio. Right? I'm not going to comment right now on this idea of anti-vaxxers and the idea of people who don't believe in vaccines, but there was a very strong marketing campaign that came along with the idea of, the polio vaccine. So, will that happen? Will we have, a mascot? Will we have a national trend? That's left to be seen. But those are some challenges that we face. The last thing I just wanted to really briefly touch upon are, now that we talked about all these vaccines, what's the alternative? And the alternative is really those therapeutic drugs. So a therapeutic drug is, again, what we're going to use to treat individuals when they actually are exposed to the virus. And so for the time being, this is a placeholder, if you will. This is sort of trying to buy us time until we actually can develop and market and distribute a vaccine. So as I mentioned at the beginning, we currently have 300 plus therapeutic treatments in clinical trials, and we'll mention a couple of them here just briefly. But there's more therapeutic clinical trials happening right now than there are vaccine trials, and I think there's some reasons for that. Some are going to be looking at new drugs and new approaches, treatments to possibly help individuals infected, and some are looking at repurposed drugs, and some are doing combinations of a lot of things. Here are just 4 basic ones that we are looking at. Number 1 is plasma donations. So that's the idea that we're using the blood from and the antibodies from somebody who has been infected, and that would be more along the lines of maybe trying to develop something new. Nitric oxide, this is actually the drug that led to the development of Viagra, and that's, relaxes the blood vessels. And as you can think, in terms of the chest and the lungs, right, if we can, use a drug that might relax blood vessels, that could, lessen some of the effects that we're seeing in these patients who are being infected. And then the last 2, maybe you've heard a lot more about. The first one is hydroxychloroquine, and so there's been a lot of political back and forth with this in the media recently. It is a drug that's approved for malaria and lupus, but the FDA did originally put an emergency authorization out that said, yes, you can use this, but they have since revoked that emergency authorization. So currently the FDA does not condone using hydroxychloroquine, and that is because at the beginning stages we had several what we would call retrospective data sets or retrospective studies that showed that it might be beneficial, so why not try it? But since when we've had some more controlled experimental design, the FDA has shown that the benefits do not outweigh the consequences of using this drug. Now there are some current things that are happening with combinations of hydroxychloroquine in experimental design, but the FDA did revoke that emergency authorization. The last one is remdesivir. Maybe you've heard of this. This is the only FDA emergency authorization treatment right now. Again, that doesn't mean it works. This is what the FDA says is better than nothing at this point. There's nothing that says that this would hurt more than it would help. And that's an antiviral drug that has been used for Ebola, SARS, and MERS. So those are therapeutic treatments. And the last thing I just wanted to leave you with is, even though we might not have a vaccine in 18 months, even though, that the acceleration might not happen, we've put all our stuff we can into the same pot, and we have a worldwide, effort, many countries working on this, researchers from colleges to, regulatory agencies to, private foundations. The HIV therapeutic data gives us hope that we can buy time. Now, this is a very old, and by very old, I'm talking April 29th. So, in the US, 60,000 dead. And it's, I didn't update this graph. I didn't make the graph, but I could probably update this now, but it still gets my point across, is that, at the height of the HIV pandemic, in the US, we had 45,000 or so deaths. And you can see as soon as we started to get combinations of antiviral drugs, that this death rate has fallen. And so even though today we don't have a vaccine for HIV, we have, quote unquote, and I don't want to even say got it under control, because the spread, isn't necessarily under control, but we have ways to treat humans that are infected with it with antiviral drugs. And so that's positive. It gives us hope that, even if a vaccine isn't happening tomorrow, that some of these therapeutic drugs will be able to strongly reduce our death rates. So that's my conversation on my talk. Lots of sources here, of course, the regulatory agencies and whatnot. And at this point, I can hopefully open up for questions. And again, I'm not a biologist, I'm a statistician, and I'm definitely not a medical doctor, but I can read a clinical trial and kind of know where we're at.
Thank you, Dr. Hassler. I'm glad my video was disabled because my jaw was probably on my floor half of the time with all the information that I was learning. So thank you.
Sorry if I went too fast.
No,
but there's so much stuff.
There is a lot of stuff, and we already have some questions coming in. So I'd like to introduce Professor Dawn Reitz, who's here to moderate our Q&A today. And I'm going to go ahead and turn things over to her for right now.
All right, thank you, Sonia. Thank you, Dr. Hassler. Wow, I agree with the jaw-dropping perspective that Sonia just indicated. I did not know that Jonas Salk was from Pitt, so there.
Don't say that.
I know, that's at least one thing that I learned. So we do have a couple of questions, and I encourage our audience to continue to submit questions in our Q&A. So, Dr. Hassler, since many who develops COVID-19 are asymptomatic, how will we know if this vaccine is effective in volunteers in the phase 3 clinical trials?
Well, yeah, being asymptomatic is one thing, but you can be asymptomatic as long as you have a test that can determine the antibodies or the virus in your body. So even though you might not have any symptoms, if these people that are getting the vaccine will be followed and will be monitored and will be tested to determine if they have been infected. Now we don't know how long antibodies will stay in the body, but it comes back to creating a test that is able to very effectively detect the presence of either the antibodies or the virus. And so we have come a long way, I believe, in 6 months in terms of the testing. There are really 2 types of testing. There's a nucleic acid test, which is the swab that you might be familiar with, and then there's the antibody test. One of them is producing rather high rates of false positives, and the other is producing currently rather high rates of false negatives. So we do have a long way to go with testing, but the answer to the question specifically is with an accurate test, we will be able to determine if the vaccine is working.
All right, great, thank you. The next question is one reason why it took so long for other vaccines to be developed due to their occurrences decreased over time, so it was difficult to find enough subjects to test vaccine candidates? If so, is it a help, sadly, that COVID is rampant in many areas of the world, especially for US, to find a vaccine and accelerate testing more than the polio, the flu, etc.?
So again, I'm not a historian, right? I'm not a biologist. So just from my research in this and from looking into, more information, I think it could be a very likely possibility. If you look at the polio, crisis, for example, I mean, it was a fairly warm weather outbreak type of scenario. So, over the summers, the outbreaks is when the outbreaks happen. And that's, to a certain degree, we had thought that maybe that would happen with this, but this is a much higher contagious rate that we're seeing. And therefore, we haven't necessarily seen it react to differences in temperature. So could it happen so I guess the question, maybe I have two sort of parts of the question. The first part is, I don't think it necessarily makes a difference in terms of the length of the length. I mean, so polio is one example, but you could look at HIV, which is, not, seasonal either. But when you put a vaccine into somebody, it's the idea of they have to be exposed. So even though, if you want to see if it's effective, at some point you need to see how the rate of infection is happening. So that could be possible, but even though some of those that we talked about, like the flu and is seasonal and polio, we also have HIV and chicken pox, that necessarily wouldn't be. What was the second part of the question?
I'm sorry.
That's my bad.
If so, is it a help, sadly, that COVID is rampant in many areas of the world, especially the US, for us to find a vaccine and accelerate testing more than polio?
So yeah, I guess, that's a unintended positive consequence, maybe, is that because it's so rampant, it's not going to be hard. So like the Moderna trial is looking at 89 sites across the US and looking at hotspots. And so there's a lot of hotspots. So, that could potentially help to the acceleration of finding a vaccine, because if we are putting the vaccine into people in these high infection areas, we might not have to wait a year or 2 years, right? So that could be, but this also is, somewhat brand new and novel to a lot of us. So yeah, I think that could be an unintended positive of, I even hate to say how contagious this is, but yeah.
Okay, thank you. Here's a question. How realistic do you think the 18-month timeline is?
So I do not, again, this is my personal opinion. I do not think the 18-month timeline in terms of a widespread distribution that would be effective to gain herd immunity is very possible.
Okay.
And now I'm not on the front lines of but I think when you look at, yes, we're doing a lot of things to accelerate, but I haven't heard a whole lot about the, ramping up of, factories to get this vaccine that still hasn't been found. I haven't heard a lot of that. Not saying it's not there, but personally, I think the therapeutic drugs is going to happen, and we'll be able to decrease some of these effects before we have a widespread vaccine.
Okay, good, thank you. We have more questions coming in. Are we certain that all the labs working on the vaccine are sharing pertinent information? I would hate to have each one reinvent the wheel. So I think that this, I was gonna say this caller, but this guest of ours is asking, is everybody playing nice? And then she says, by the way, Dr. Fauci just mentioned that he had high hopes for a vaccine by the end of 2021.
Yeah, by the end of 2021. Right. So now hopes of a vaccine doesn't necessarily mean that you and I would be able to get that vaccine. All right. So, do I think that we could have potential vaccine candidates that we have studied for, 12 months or so that seem to be effective? Yes, I think that could happen. But in terms of the widespread distribution and whatnot, I don't know. Again, that's my personal opinion. But the other part of the question was, is everybody playing nice? Again, I'm not on the front lines. I don't know that, but that's part of what the World Health Organization does, right? They're tracking 70 of the 200 plus candidates right now, which, are, the World Health Organization is developed to try to help to collaborate across, countries and across the world. I would say we have so many more people, so much more money put into this than other vaccines that we have seen in the past, and because this is happening worldwide, and so it's a lot better than what it used to be, I would say, is what I'm seeing.
Good. Thank you. More questions. You mentioned the polio vaccine was about 80% effective. If COVID is more contagious in comparison, will the vaccine require a higher effectiveness? Then, is there a direct relation between the disease's rate of infection and a vaccine's effectiveness that result in disease suppression?
So that's a lot.
In terms I can ask it again if you need.
No, it's fine. In terms of, again, I'm not a, pharmacologist. I'm not a, infectious disease expert. But I can look at the numbers and I can look at the math and the probability, right? So my initial response is that it goes back to the herd immunity. I can't even talk. Humanity, right? So the idea that, if we really do need 50 to 67% of the people that either have had the disease or have vaccinated, then you don't necessarily need an 80 to 90% effective vaccine. So if you look at the probability, right, even if we have a 50% effective, if we're giving that to a large portion of the population, we could potentially start to push that herd immunity, up higher. So again, that's probably, if you look at the mathematics, the probability behind it, and a lot of that is going to determine, even though I mentioned we're analyzing statistical data, there's not a whole lot of data to analyze yet. Right. So, maybe 3 months from now we can have another LionSide chat and I can show you results of stage 3 trials. Right. But we don't have a whole lot of data yet. We just had those beginning stages.
Awesome. Well, being one of the LionSide chat moderators, I will hold you to that three months.
Oh no, what did I just put myself into?
Another question from our audience. Do you think the quality of the vaccine will be compromised from the hurry to get it on the market?
Do I think it will be compromised? It just depends, right? The FDA, has repetitively said that they're gonna, stick to the standards. So I guess it just depends on what is happening and those decisions, that are being made. I think, unfortunately, especially if it's coming from the U.S. with, our political unrest, that, I would hate to see, that get involved. But, from the FDA, we have these things in place. We don't want to let something get into the, community that would, hurt us more than help us. And the people who are monitoring these, peer-reviewed journals, the people that are on the FDA, panels for approval, are not just government officials, right? There are scientists, there's pharmacologists, there's market managers, there's clinical trial, specialists. So I think it could. Mm-hmm. It's yet to be seen if it will.
All right, good. Maybe more in our 3 months from now, LionSide Chat on that. Another question here, and if you have questions, audience, please feel free. We could probably take another 2 or 3. In regard to therapeutic drugs, Dr. Hassler, do you feel that with our current death rate, we should be using retrospective studies to approve potential treatments in order to speed up options to treat?
Yes, retrospective studies, and I was anticipating this question might come up, but retrospective studies are not bad if you have no, a retro, for those of you who aren't familiar with it, retrospective data is looking in the past. It's not setting up with a controlled design. It's not looking at what is happening in the future and monitoring things. It's looking at the past. And so if we do have, past data to analyze and we have nothing else, good. Right? Let's start with that. That's why hydroxychloroquine was emergency approved at first, because we looked at some past data. But the idea is you can't really set up the experimental design. You can't look at what person, you can't separate groups. You don't know what pre-existing conditions necessarily. And if you do, you have no control over, affecting or switching. So, yes, it is a, I know I've done a little bit of research on that in the last couple of weeks. It is common in clinical trials. But in terms of moving forward, we now have more, I, as a statistician, would argue better evidence from our experimental designs that suggest that they, hydroxychloroquine is not an appropriate drug, and therefore the FDA has, pulled back their emergency authorization of it.
Okay, thank you. And just as a reminder, audience, you're welcome to submit questions to our Q&A.
So Dr. Hassler, I'm going to go next. When a vaccine is found, okay, how will the general public know that it is safe and effective?
Yeah, I anticipate that it could be very politicized. The media, we have 2 extremes in terms of the media. That's not I don't think too many people would argue that. But, it's the idea of, educating yourself on the process. What we, thank you for joining us today here is to give you a couple, points of what's happening. But, first off, is it peer reviewed? Right? Has it been FDA approved? Right? Even though the Federal Drug Administration is the approval process for the United States, it could very well happen that a vaccine comes out of Europe or a vaccine comes out of China first. And so, you'd have to look at the regulatory, processes of those places. Of course, the FDA, if we're going to use it in the United States, would have to approve it as well. But has it been peer reviewed? And then again, looking at the things we've talked about in terms of the acceleration of the timeline. But I just go back to those 3 principles of scientific research, the randomization, the replication, and the control. And so, yeah, I've been educated on how to read clinical trials, as of course the general public probably isn't, but I would just argue research, and, look into it and think about, the processes. And as I said earlier, maybe come back for another LionSide chat if we have more information.
Okay, great. Thank you.
Yeah, I think that's an interesting concept, and it's definitely something that I know in my circles that are not statistical at all. We discuss this. If there's a vaccine, are you going to take it? We do have another question. Branching off of that question, there are many people who respond to the virus differently. Even if the drug is tested on a surplus of subjects, how are they so sure it will be effective on the masses? How much would the different symptoms of the disease alter the effectiveness of the vaccine?
Yeah, so I guess one comment I might have, and again, I tried to not put my opinion in here too much, but with how much we have at stake and in sort of in the game here, I don't know. I can hypothesize that we might have more than one vaccine, and we might eventually have more than one vaccine that could be targeted for different individuals. So there might be a vaccine that could be used for elderly versus pediatric. I could see that could possibly happen. But the point of the question, to answer what the participant just asked here, is that if your stage 3 clinical trial represents the population that you're going to be putting the vaccine into, then that gives you evidence that it would be, spread, a countrywide or worldwide, applicable to that population. Now, if we start to fail on these clinical trials and don't see a lot of good evidence happening, then you might start to see, cohorts of specific type of people, adverse reaction, elderly people, pediatrics, and we might start to look for, vaccines that could be effective in those, sub cohorts of people.
All right, great, thank you. And it looks like we have time for one last question here, so here it goes. Does the FDA redo the phase 1 and 2 studies that the original research performed, or just double check them somehow?
I mean, it's a regulatory agency. So the FDA is not in charge of running the clinical trials, right? So the clinical trials are done by your pharmaceutical companies. They're done by your research institutes, your private foundations. So, I mean, I'm not saying that the FDA might not say, hey, you need to go back and replicate this and we need to modify and change stuff, but they're the regulatory agency that moves the drug or the vaccine into the next stage. And so I guess, I hate to put a blatant answer out there as no, but yeah, I mean, the FDA isn't the person who's going to be running the clinical trials. They could be providing recommendations, tell a candidate that, no, you have to pause and redo this and change this, and then come back with us with your, safety and efficacy results.
Great. Thank you so much, Dr. Hassler. We are wrapping up. It has been the fastest hour of our lives here. No, this is great. We'd like to thank you for chatting with us today and sharing, some information about the upcoming phases and what we really need to go through before we get a vaccine that works and maybe even as a bonus tastes good, like polio. So we would like to also thank Stanley, our tech tutor, for standing by today if we had any technical difficulties. And a special thank you to our audience for joining us for today's LionSide Chat. As you click out of the webinar, you're going to receive access to a survey, and just take a few minutes to complete the survey to let us know what you thought about today's chat and maybe even offer some ideas for future chats with Dr. Hassler or even some other members of our campus community. So we welcome you to reach out to us via email to let us know if there's any other topics that you're interested in learning about. And we would like to also let you know that we're check our website for any upcoming LionSide Chats. As you can see, our next chat is this Wednesday, August 12th, where we're going to be talking about the impact of COVID-19 on the drug overdose crisis here in the United States. So be sure to come back soon, meet with another faculty, staff, or student from Penn State Berks to share their experiences. And most importantly, stay safe, Berks and beyond. Signing off until next time, this has been the Penn State Berks LionSide Chats.
Thank you.